Right now, the world is fighting a disease called coronavirus disease or COVID-19, and it was caused by a newly virus called Severe Acute Respiratory Syndrome Coronavirus 2 or SARS-CoV-2. According to Kompas News, on 3rd May 2020, the number of reported cases in Indonesia has jumped to 10,843 cases. Now, let's take a look at its general structure. It is an RNA virus that has an envelope. It is also a positive sense virus and highly diverse group virus. Scientists have used two techniques that are fast and accurate to detect this virus. It was called rapid test and RT-PCR. The most common rapid diagnostic tests that have been marketed globally is rapid test. This rapid test kit is able to detect the presence of the antibody in the blood samples of people that may have been infected by the coronavirus. However, this RDT has drawbacks because it may give false positive results. Thus, WHO does not recommend the use of antibody detecting rapid diagnostic tests for patient care. Then, what other methods that we can use? We can use the real-time reverse transcriptase polymerase chain reaction, or for short, real-time RT-PCR. This method can detect the specific genetic materials like DNA or RNA, including viruses. This method uses reverse transcriptase as a tool to change the RNA molecule to DNA and amplify those DNA using polymerase chain reaction. To monitor the amplification process, the fluorescence is being used in this method. And from that, we can raise one question. How exactly RT-PCR detect the virus genetic code? To detect the virus genetic code, we need to know the structure of the coronavirus itself. As mentioned before, the coronavirus is an RNA virus. So, coronavirus has the RNA molecule as its genetic code. Apart from that, it has the S protein, the RNA genome, the envelope, the E protein, the N protein, and the M proteins, which all are accessory proteins that are essential for the viral survival. The most important protein that the virus needs for its survival is encoded in the ORF1A and ORF1B, which function for the virus replication process. There are two target genes identified in the ORF region, which are RDRP gene and E gene. Apart from that, we can also use N gene for the identification process. Now, apart from here, we can raise one question again. What exactly is the RDRP gene and the N gene function in the virus? The RDRP gene is a gene that produces enzyme to replicate viruses when infecting human cells, while N gene is a gene that protects the core of the RNA virus. To detect these two genes, the suitable primer could be chosen based on this table. There are three single steps to collect this virus. 1. Obtaining the clinical samples. The clinical samples are collected from both the throat and nose of the patient, which is called as a swab test. The sample was then treated with the PBS buffer and then vortex to transfer the virus to a new media, and then the supernatant was taken. The second step is RNA extraction. It was started by adding the binding buffer to the samples, the RNA binding, and adding the washing buffer twice, drying, add the elution buffer, and then we got the viral DNA. The third step is the RT-PCR. In this step, the RNA will then be transcribed into the cDNA with the help of a specific enzyme known as RNA polymerase. The scientists will also add specific primers that are complementary to the transcribed DNA. These primers are used for the binding to the DNA strand and for adding the marker label to the strand to detect the virus. Okay, now let's take a look at an example. It was started by the target and primer selection. In this video, we will have two kinds of assay, but for the example alone, we will use the assay 1 as the example, which target the ORF1b as the target. Next, we prepare the master mix for the RT-PCR. It consists of the RNA-free water, Tagman master mix, forward primer, reverse primer, probe, and samples. After that, we set the RT-PCR machine to several conditions. First, we set the machine in 50 degrees Celsius for 5 minutes. In this step, the reverse transcription process is happening, and it converts the RNA molecule to complementary DNA or cDNA. Next, we set the machine to 95 degrees Celsius for 20 seconds. In this step, the initial denaturation is happening, which activates the tag polymerase to prepare the PCR process. Okay, now we move on to the PCR process, which has 40 cycles. Set the machine to 95 degrees Celsius for 5 seconds, which in this stage, the denaturation process is happening. It separates the DNA into two single strands. Finally, we set the machine to 60 degrees Celsius for 30 seconds. In this stage, the annealing and extension process is happening. It enables the primers to attach to the template DNA and building the new strain of DNA by tag polymerase enzyme. And now we wait. As we can see here, the white bar is the control group, while the blue bar is the patient sample. The level of E protein and the RDRP gene in the patient is higher than the control. By this difference, we can know whether the person is infected by coronavirus or not. For all listeners at home, we have terrific news. There's a news coming from Indonesia regarding the successful production of a prototype for RT-PCR COVID-19 test kit in Indonesia. The test kit is called Nusantara TFRIC-19 and is being developed by Nusantics. The design was made according to the genomic data of the virus spread throughout Asia. This test kit was produced by Nusantics, will be produced again massively by BPPT, Bio Farma, and Indonesia International Institute for Life Science or I3L. So, what is the coronavirus strain that we use to detect the COVID-19 in Indonesian patients? Do you want to know? Apparently, Indonesia has already used their own strain to detect this disease. I3L has contributed many things in the process. They provide us a laboratory and also a volunteer for the RT-PCR kit development. For instance, they have a laboratory with the HEPA filter and BSC. Also, they provide a credible volunteer from the I3L student itself. However, we still want to say thank you very much for I3L and all medical professionals that help fighting in the front line. But it's not only them that can help fighting this disease. You can help. Yes, you. Please check out the links as shown here where you can send your donation to help fight the COVID-19. That's all for today. Thank you for watching our video. Don't forget to wash your hand and see you next time. Bye-bye.